Induced pluripotent stem cells represent another type of stem cells. They are pluripotent just as embryonic stem cells. Thus, they are able to generate every cell type found in our body, such as neurons, gut cells, or muscle cells. They are obtained from differentiated cells upon infection with a virus that activates expression of 4 particular genes. For this seminal discovery, the Nobel Prize in Physiology has been awarded to the Japanese scientist Shinya Yamanaka in 2012. Induced pluripotent stem cells have potential advantages with respect to the other types of stem cells. Differently from embryonic stem cells, they do not pose ethical concerns since they are not derived from embryos. They can be obtained simply by blood sampling. They are suitable for autotransplantation. Indeed, differentiated cells can be collected from a patient, induced to the pluripotent state, differentiated into a specific cell type, and then transplanted, avoiding most of the complications of rejection. The main problem that has to be addressed in order to use induced pluripotent stem cells for therapy is their safety. The time interval to induce the pluripotent state and the differentiation of cells to be transplanted into the patient is quite long, and this can lead to the occurrence of mutations that can be transferred to the patient. For this reason, induced pluripotent stem cells are not yet used in clinical practice. To date, only one clinical trial has been opened and then suspended due to the presence of a mutation in the reprogrammed cells. At the moment, induced pluripotent stem cells are used to create huge collections of stem cells called biobanks. They are derived from people from all over the world and can be potentially used for any type of transplantation. They are widely used to study rare genetic diseases. One example is progeria, a very rare disorder that affects one child every 20 million, characterized by premature aging. Patient cells are reprogrammed, and since they carry genetic alternation that causes the disease, they are used to reproduce it in vitro in order to study the underlying molecular mechanisms. Induced pluripotent stem cells represent an important scientific discovery, although they have quite a long way to go before they will be actually applied in clinical practice.